This protocol provides a robust, reproducible model of α-Synuclein accumulation in primary dopamine neurons for studying the mechanisms and treatments that may regulate this accumulation. Preformed α-Synuclein fibrils induce progressive, highly reproducible protein accumulation. Combined with automated imaging and unbiased image analysis, this protocol facilitates a relatively fast, medium to high throughput screening of α-Synuclein accumulation inhibiting drugs.
Progressive α-Synuclein accumulation may be one of the factors compromising neuronal function in Parkinson's disease and synucleinopathies. New molecules blocking such accumulation may become new therapies for neurodegeneration.
We believe that this protocol is an important step for establishing a standard and reliable tool to study molecular mechanisms regulating α-Synuclein accumulation in dopamine neurons. Demonstrating the procedure will be Yulia Konovalova, a PhD student from my laboratory.
Before setting up the primary embryonic midbrain cell culture, add 50 microliters of dopamine neuron medium to each well of a poly-L-ornithine-coated 96-well plate. And use a 100 microliter pipette tip to aspirate the medium, while simultaneously scratching the bottom of each well in a circular direction to remove the coating at the perimeter of each well. A poly-L-ornithine-coated island will remain in the middle of each well. Add 10 microliters of medium to the middle of each coated island to create microislands. To set up primary embryonic midbrain cultures from mouse embryonic day 13.